Consider the vastness of space. Now consider something even more awe-inspiring, the intricate universe contained within each of us. Our brains, those 3-pound universes of fleshy matter, hold the keys to our thoughts, our emotions, our very being. Billions of neurons, interconnected in a complex web, communicate through a language of electrical and chemical signals. This intricate dance of ions and molecules allows us to perceive the world, to learn and remember, to dream and aspire. This essay embarks on a journey into this inner cosmos. We will delve into the fascinating world of neurons, the fundamental units of the nervous system. Our exploration will uncover the secrets of their communication, the electric messengers that race along neural pathways carrying information throughout the body. From the simplest reflex to the most profound thought, it all boils down to the language of neurons. Even the simplest organisms exhibit a fundamental characteristic: irritability. This ability to respond to stimuli is essential for survival. Consider the humble amoeba, a single-celled organism inhabiting ponds and puddles. Despite its simplicity, the amoeba can detect and react to changes in its environment. It can move towards food or away from danger. This basic responsiveness hints at the foundation of our own nervous system. In multicellular organisms like ourselves, specialized cells called neurons have evolved to carry out this task of communication. Neurons are the fundamental units of the nervous system, transmitting information throughout the body via electrical and chemical signals. This intricate network of neurons enables us to perceive the world around us, to think, to feel, and to act. Neurons, the building blocks of our nervous system, possess a unique structure well suited to their role as information messengers. Imagine a tree, its branches reaching towards the sky. The neuron shares a similar architecture. The cell body, or soma, forms the neuron's core, housing its genetic material and cellular machinery. Extending from the cell body are numerous branch-like structures called dendrites. These dendrites act as receivers, picking up signals from other neurons. A single long fiber called the axon stretches out from the cell body, resembling a trunk extending from the tree. The axon serves as the neuron's transmission cable, carrying electrical impulses away from the cell body towards other neurons, muscles, or glands. These signals, known as nerve impulses, are the electrical messengers that allow neurons to communicate with each other and with the rest of the body. Imagine a neuron at rest, poised like a runner at the starting line. Even in this state of apparent quietude, the neuron is not truly at rest. Across its membrane, a difference in electrical charge exists, a delicate balance maintained by the constant movement of ions. This difference in charge, known as the resting potential, prepares the neuron to fire like a prepped electrical circuit. The neuron's membrane acts as a gatekeeper, selectively allowing certain ions to pass through while restricting others. Pumps embedded within the membrane, such as the sodium-potassium pump, work tirelessly to maintain this ionic balance. This pump actively transports sodium ions out of the neuron while drawing potassium ions in. This continuous pumping action creates an electrochemical gradient across the membrane, a difference in both charge and ion concentration. The resting potential is a testament to the neuron's constant vigilance, always prepared to respond to the next incoming signal. Section 5, the electric wave, the action potential's journey. When a neuron receives a sufficiently strong signal from its neighbors, it doesn't merely whisper its message, it unleashes a surge of electrical activity, a sudden and dramatic shift in its membrane potential known as an action potential. This electrical wave, a cascade of depolarization and repolarization, races down the neuron's axon, carrying information to its destination. The action potential is an all-or-nothing event, like a digital signal. It either fires at full strength or doesn't fire at all. This ensures the fidelity of the signal as it travels long distances along the axon. During depolarization, voltage-gated sodium channels in the membrane fling open, allowing a rush of sodium ions into the neuron. This influx of positive charge pushes the membrane potential towards a positive value, like a wave cresting. Repolarization follows swiftly as voltage-gated potassium channels open, allowing potassium ions to flow out of the neuron. This outflow of positive charge returns the membrane potential to its resting state. The action potential doesn't simply fade away, it continues to propagate down the axon, like a domino effect, ensuring the signal reaches its intended target. Section 6. Leaping the gap. Unveiling the secrets of synaptic transmission. Neurons, despite their intricate connections, do not actually touch each other. A microscopic gap, known as the synapse, separates them. Here, at this junction where neurons meet, the language of the nervous system transforms from electrical to chemical. The arrival of an action potential at the axon terminal triggers a remarkable chain of events. Voltage-gated calcium channels open, allowing calcium ions to flood into the axon terminal. This influx of calcium triggers the release of neurotransmitters, chemical messengers stored in tiny sacs called vesicles. These neurotransmitters diffuse across the synaptic cleft, the narrow space between the neurons. Upon reaching the postsynaptic neuron, they bind to specialized receptors like keys fitting into locks. This binding initiates a cascade of events that can either excite or inhibit the postsynaptic neuron, influencing its likelihood of firing an action potential. Section 7. Shaping memories, synaptic plasticity, and the learning brain. Our brains are not static entities. They are constantly changing, adapting to new experiences, learning and remembering. This remarkable ability of the brain to modify its connections and function over time is known as synaptic plasticity. Like a sculptor molding clay, experience shapes the connections between neurons, strengthening some synapses and weakening others. Synaptic plasticity lies at the heart of learning and memory. When we learn something new, new connections are formed between neurons and existing connections are strengthened. This process of synaptic change alters the flow of information through the brain, allowing us to store and recall memories. Long-term potentiation, LTP, a persistent strengthening of synapses, is thought to be a key mechanism underlying learning and memory. LTP involves an increase in the strength of synaptic transmission, making it easier for neurons to communicate with each other. Section 8. When signals go astray. Drugs and neurotransmission interference. The delicate balance of neurotransmission is crucial for normal brain function. However, various substances, both medicinal and recreational, can disrupt this delicate dance of chemicals in the brain, altering mood, perception, and behavior. Drugs can exert their effects at various stages of synaptic transmission, from neurotransmitter synthesis and release to receptor binding and reuptake. Antidepressants, for example, often work by increasing the levels of certain neurotransmitters in the brain, such as serotonin and norepinephrine, which are involved in regulating mood. Cocaine, on the other hand, exerts its stimulating effects by blocking the reuptake of dopamine, a neurotransmitter associated with pleasure and reward. This leads to an accumulation of dopamine in the synapse, amplifying its effects and producing feelings of euphoria. Understanding how drugs affect neurotransmission is crucial for developing effective treatments for various neurological and psychiatric disorders. Section 9. A symphony of signals, concluding thoughts. From the simplest reflex to the most complex thought, the nervous system relies on the intricate communication between neurons. These remarkable cells, with their specialized structures and electrochemical signaling mechanisms, form the foundation of our perceptions, our actions, and our very consciousness. The journey into the neurons world reveals a universe of complexity and elegance, a testament to the power of biological evolution. As we continue to unravel the mysteries of the brain, we gain a deeper appreciation for the symphony of signals that orchestrate the human experience.